What if hydrophilicity isn't something we directly predict anymore? From simple scales like Parker and Kyte-Doolittle, to machine learning, to deep learning, and even AI that understands proteins like language, this field has completely transformed. Today, we don't just score amino acids, we predict structure, surface, and context. Let's quickly understand classical protein scales used for epitope and surface prediction. First, the Kyte-Doolittle scale helps identify hydrophobic regions, especially transmembrane segments. Hopp-Woods and Parker focus on hydrophilicity, highlighting regions that are likely exposed and antigenic, making them useful for B-cell epitope prediction. Then Emini and Janin shift the focus to surface accessibility, helping us find residues that are likely on the protein surface. All these methods use sliding windows, so they're fast and easy to compute. But they have limitations. They don't consider 3D structure or long-range interactions. What if we could teach a computer to find epitopes for us? That's where machine learning comes in. BepiPred learns from real experimental data to predict antibody binding regions. ABCpred uses neural networks to detect hidden patterns that simple rules can't see. So instead of fixed scales, we now have models that learn. But there's a catch. They're only as good as the data they're trained on. What if we could teach machines to read biology like a language? That's exactly what deep learning is doing today. Modern approaches go beyond simple rules. They learn patterns directly from massive biological data. They combine sequence, structure, and context to make smarter predictions than ever before. From epitope prediction to protein folding, deep learning is now the gold standard. What if seeing a protein in 3D could reveal exactly where the immune system attacks? That's the power of structure-based methods. Instead of just reading sequences, these approaches analyze the actual shape of proteins, identifying exposed, accessible regions where antibodies can bind. Structure turns hidden biology into visible targets. What if proteins had a language, and we could actually understand it? Protein language models treat amino acid sequences like sentences, learning patterns from millions of proteins. They capture hidden rules about structure, function, and evolution, without needing explicit labels. Models like ESM can predict folding, interactions, even mutations.